Hey, before we go into enzymes and chapter 8, I just wanted to go over something about the isotonic, hypertonic. So let's say you might have done this in your lab. I just want to kind of reiterate it. Let's say you have a U tube that looks like this, with a semi-permeable membrane in the middle right here. And let's say you have 0.8 molar sugar or glucose here, C6H12O6, and you have 0.2 molar NaCl, which is sodium chloride with common salt. And on this side of the tube, you have 0.6 of sugar and 0.6 of salt. Okay. So the total molarity of the solute here is going to be 6 molar for the sugar. And because it's sodium chloride, and sodium chloride, when you add it into water, it breaks up into Na plus. It'll break up into Na plus because it's ionically bonded into Na plus Cl minus, because it breaks up into Na plus and Cl minus. So you have 0.6 of this and 0.6 of this. So then that's 1.2. So the molarity here is 1.8 molar. Just the solute concentration there is going to be 1. This one is not going to break up because it is covalently bonded, and in water it's not going to break up. On this side, then you have the same thing, 0.2 of Na plus and 0.2 of Cl minus. So you're going to have 1 point, this is 4, so that's 2 molar. So this side is going to be more hypertonic, like it's going to be hypotonic to this side. Talking about enzymes, as we talked about in the past, enzymes are actually biological catalysts. In a chemical reaction, when you have a catalyst, the catalyst hastens up the reaction, but does not actually take part in the reaction itself. For any reaction to take place, there has to be collisions in the right orientation. So whether it's an endothermic reaction or an exothermic reaction, you need to provide it a certain amount of energy, which is what this is. The top part of this is the amount of energy you need to provide to it, and that is called the activation energy. When you have an enzyme, the enzyme actually reduces the amount of activation energy required. So you're saving about this much amount of energy, like as you can notice here, you're reducing the amount of activation energy. And enzymes, which are biological catalysts, can be used over and over and over again. The substrate attaches to it, the reaction takes place, and then it lets go, and then the enzyme moves on to the next product. The next. So in a reaction, we always know what, in a reaction, there's going to be 2 parts to a reaction, as you guys all know this. So there is, let's say, A plus B is going to give you C. This side of the reaction is a reactant, and this side is the product. So reactants and products. If the reactant that needs an enzyme that acts on an enzyme, if there's an enzyme that acts on it, then that type of a reactant is called a substrate. The enzyme basically will attach to the substrate, and that is called a enzyme substrate complex. So a good way to think about it would be all substrates are reactants, but all reactants don't necessarily have to be substrates. Enzymes have something called as an induced fit. A very rudimentary way of thinking of it would be, think of it as a lock and key model. This is a very rudimentary way of thinking of it. It's just to give you kind of a mental image of what it is. And I'm going to go to an image of it, and it's a lot easier that way. Let's see if so every enzyme has something called as an active site. The active site is going to be exactly the shape and has the same conformation that allows for a unique substrate to fit in it. For example, an enzyme called lactase, ASE, has an active site that uniquely fits a lactose, which is found by the combination of glucose and galactose with a 1,4 glycosidic bond. So the enzyme that is lactase will uniquely fit the lactose the substrate. The substrates come in, they attach themselves to the active site, so they kind of lock into the active site, and then the substrates are in that area are converted into products. For example, if we were taking the same example of lactose and lactase, the lactose molecule comes in, locks in there, and then a hydration reaction will take place. And once the hydration reaction takes place, the glucose and galactose, the 1,4 glycosidic bond is broken, and then the glucose and the galactose, since their shape is different from the shape of the lactose molecule as is, it doesn't fit into the active site anymore, and then it is released. And once it's released, the active site is vacant again, so the next glucose lactose molecule can come in and fit in there. And this pretty much, when you look at all the different macromolecules that we talked about, almost all of them will require some form of enzyme to break them up. Can they break up by themselves given time and enough amount of energy? Yes, they can. But the point of having an enzyme is to speed up these reactions and make them metabolically available. Enzymes are proteins, so they are very much affected by environmental factors such as the temperature as well as pH. When we talked about proteins, we talked about how there can be, if the temperature range or the pH range changes, proteins can very easily be denatured. They function best in a certain range. So for the proteins that are in your blood function very well at body temperature, within a range of body temperature and a pH of 7.4. Any change in that would actually start denaturing the proteins. This is why we have the process of homeostasis in order to maintain the pH balance of your body and maintain the temperature of your system. There are a lot of different types of chemicals that can also specifically impact your enzyme. It can impact the active site. It can change the shape of the active site. It can even lock the active site. It can be competitive and can lock the active site. So the actual enzyme, that the actual substrate that's supposed to go in there doesn't go in there. So we'll look at a couple of examples. So as you can see in this graph, you'll notice that the optimal temperature for enzymes in the human body is right here at 37.5 degrees. Their activity is the rate of their activity is much lower, and then makes a sharp decline as it goes further. Whereas thermophilic bacteria, thermo meaning heat, philic loving, bacteria that are heat tolerant or love the heat, tend to have an optimal temperature of 80 degrees. So So a good way to think about it also is that bacteria that are heat tolerant, organisms that are heat tolerant, like a thermophilic, can actually not make you sick, because if you look at its activity, first of all, at 37 degrees, there is zero activity in its enzymes and its proteins. And even if you had like a fever and your temperature went all the way up to 40 degrees, it would be very dangerous. Even then, it has very, very, very little activity. So if it was to be introduced into your body by some means or the other, there is no way it's going to make you sick, because it is literally not going to be able to function in your system. The proteins tend to get denatured at that point. At the same way, you'll notice that there is, with acids, the pH in your stomach is between 1 and 3, and its optimal functioning range is at 2. So pepsin, which breaks pepsin, the enzyme pepsin would break peptide bonds. So the names of the enzymes pretty much will tell you what the substrate it's working on is called. It's going to break down peptide. So pepsin is released in the stomach, and it functions by breaking down peptide bonds in the stomach. Peptide bonds are found between 2 amino acids. In the small intestine, it is released by your pancreas in a non-functional form called trypsinogen, and then it is activated in your small intestine and becomes a trypsin. Trypsin also breaks down proteins, but this is only found in your small intestine. If you were to transfer this enzyme to this location, it's going to denature and not work. If you're going to transfer this enzyme to this area, it's not going to work. So this is what we mean by the enzymes have a very specific range of temperature and pH. Besides the substrate, there are other non-protein enzyme helpers that kind of maintain, that help in maintaining the shape. They help in activation of the enzyme. They also protect. There are sometimes where they kind of protect the active site, and the inorganic ones are usually metals, like when we say metals in their ionic form, we're talking about calcium ion, Ca+2. It can be an iron, which would be Fe+2, zinc, Zn+2. If it is an organic cofactor, then we call it a coenzyme. Coenzymes almost always include vitamins. That is the role of vitamins. If you think back to chapter 2 or 1 or something like that, we talked about some things you need in trace elements. You need a small amounts of it. Vitamins are one of those things because they function as coenzymes. Metals, heavy metals like iron, phosphorus, manganese, calcium, you need them in small amounts because they also function as cofactors. So as we talked about it, we know that the substrate is very unique to an enzyme. So sometimes there can be a competitive inhibitor. Something else might lock into the enzyme's active site, rendering it useless for whatever function it was performing. For example, let's take hemoglobin. Hemoglobin is an enzyme in your blood that carries oxygen, that carries oxygen through the blood. One of the competitive inhibitors, that's the one that locks into the active site, is carbon monoxide. Carbon monoxide is a competitive inhibitor. So if you have carbon monoxide poisoning, that means there has been a large concentration of carbon monoxide. You've inhaled a large concentration of carbon monoxide, and it starts locking your hemoglobin. It's competing for that active site instead of oxygen. So that then the amount of oxygen being transported to your body reduces. If this happens and you go to the hospital, basically this is a reversible situation. So what they'll basically do is increase the concentration of oxygen in your body, and if they increase the concentration of oxygen in your body, then it is more competitive than carbon monoxide. So then the hemoglobin picks that up because there's a concentration difference. The next type of inhibitor is a non-competitive inhibitor that binds to another part of the enzyme that's causing the shape change. So, for example, if I was to, like, there might be an image. So in the case of a non-competitive inhibitor, so this is a competitive inhibitor. This is the one, let's just look at all the pictures. Here's the active site and here's the substrate. Normal binding would be that this attaches to this. A competitive inhibitor might be this one where it's attached to this area, like we talked about carbon monoxide, and then the actual substrate is not able to attach itself. So the way you would remedy this is by increasing, by Le Chatelier's principles in chemistry in terms of volume, and you would just increase the substrate concentration, and you could in many cases reverse it. In cases like cyanide, it's very difficult to reverse it, so you may not be able to reverse that. Now in the case of a non-competitive inhibitor, a non-competitive inhibitor is not attaching itself to the active site. Instead, it's attaching itself to a different site, which is an allosteric site. And by attaching itself to to a different site other than the active site, it kind of changes the conformation. Every time we use the word conformation, we are talking about shape. It changes the shape of the active site itself. So since the shape of the active site has changed, now the substrate cannot attach itself to it unless something removes that competitive a non-competitive inhibitor. Now this allosteric site does not always have to be something that's detrimental, that's going to kill you. It's sometimes used by the body to do regulation. So if there is no need for an enzyme to be functioning, then it can create an inactive form of that enzyme by locking it at the allosteric site and changing its conformation. And we'll do a lot of this this kind of changing conformation as we do translation and transcription, and when we talk about different ways in which transcription and translation is regulated. So a good example for a non-competitive inhibitor that can also be very dangerous is a strychnine, which blocks the path of glycine. Allosteric regulation. Allosteric regulation of enzymes is where the activity of the enzyme is actually affected, either stimulated or inhibited. So it's actually affected by some sort of a molecule that binds to the protein at a different site, a regulatory molecule that binds to the protein at a different site, changing its conformation, changing its functionality, because of which it can either be turned on or turned off. This is a way in which your body makes sure that enzymes are only working when they are required to work. These enzymes require a lot of energy to be made. So the body tends to regulate its use. If there is no need for it, there is no point in having it in the system. For example, there's no need to have trypsin in your small intestines if there is no protein in it, if there is no digestion taking place. So an inactive form is is available in your pancreas. There is no need to keep it in the form of trypsin to make it in the form of trypsin. So a trypsinogen exists. Similarly, in your system, there's no need to have pepsin just all over the bloodstream. It might land up attacking other proteins and breaking them down. Instead, you have an inactive form called pepsinogen. So pepsinogen is found in its inactive form, so it's not actually creating any chaos by digesting things it shouldn't. So just like we talked about, that enzymes have both an active and inactive form. Enzymes also have an active, sometimes they can have an activator that kind of stabilizes its function. It can sometimes stabilize it by inactivating it. You really don't want enzymes to be all just running around willy-nilly, because if they find their substrate, they are going to break it down, and you have to have a system of controlling it. Image of that shows how something is stabilized to be active as an activated form or an inactivated form by using separate molecules. So allosteric regulators are very are very good drug candidates. Most of your cancer drugs are based on allosteric enzymes. So they basically inactivate. Cancer is uncontrolled mutation and growth. It's growth without differentiation. So your cells are continuing to grow, but they're not differentiating to do the job that they're supposed to do. And they are also immortal cells because they continuously keep reproducing. So you want, and there are a ton of proteins involved in this reproductive in this uncontrolled reproduction of cells. So you can have a ton, most of your cancer drugs are going to be allosteric regulators that kind of lock the proteins that are required for replication and then make them and convert them into an inactive form, so then they can inhibit replication. They cannot inhibit replication completely, because then that's going to cause you not to be able to replicate healthy cells. So they would actually reduce replication in certain number of cells. They're quite targeted, and then they reach the area that they're supposed to reach, and they stop replication by actually inactivating the proteins involved in it. So this is the location. This is the active site. This is where, just to recap what we've already done, this is the active site. The substrate is going to attach here. This is going to be your allosteric site. In the allosteric, as you change the shape of the allosteric site, the shape of the active site is changing. So let's say this was an allosteric inhibitor. If the allosteric inhibitor inserts itself here, the whole thing is going to move in that direction and kind of change the shape of the active site. And once the shape of the active site is changed, the substrate cannot attach to it anymore. So unless you remove this allosteric inhibitor, you are not going to be able to then that enzyme is incapable of doing its function. So that would make it an inactive form. Kind of have a good idea about these things, because in this from this chapter you obviously don't have a lot of questions. The only questions you'll get from this chapter are, what is metabolism? Knowing what metabolism is. Metabolism is a combination of anabolism and catabolism. Understanding what is an enzyme, what are substrates, what is activation energy, what is the function of an enzyme, what is an active site, what is an allosteric site, a cofactor, a coenzyme, what is the function of allosteric inhibitor, what are competitive inhibitors. So most of your questions on this chapter is going to come from this last section of the chapter. You also will have to know this, kind of understand the functionality of enzymes, because as we go forward into cellular respiration, photosynthesis, transcription and translation, and all of those things, I'm going to assume that you have understood this portion of the material and just talk about it much faster. So we will be picking up a lot more speed in the next few classes, because I wanted to spend a lot of time on the chemistry portion of it and on the basics of it, so that I'm not just rushing through everything and you guys are not just like lost. So in the production system, there's always a feedback initiation. There's a feedback loop that happens. There are two different, so that the body doesn't actually start wasting a lot of its resources. So there are two different types of feedback loops that happen. One is negative and the other one is positive. Negative is the most popular and the most often occurring system, because the point of a feedback loop is to produce as many chemicals as chemical resources as required and not to waste resources. There are two hormones that actually signal for positive reinforcement feedback loop. A positive, and that is oxytocin and prolactin, and these are used in oxytocin plays a huge role in childbirth and to extend the contractions. Prolactin allows for production of milk. So let's look at a feedback loop that's a negative feedback loop. Basically what's happening is, let's assume that you need to produce, forget about the name, you need to produce an end product. Let's call this end product AB. So to produce AB, first an intermediary product of A is produced, and then an enzyme comes in, B, C, D is produced. As the concentration of AB increases in the system, if there is more and more AB in the system, this AB will then go ahead and bind to the allosteric site and change the conformation of the active site. And once it changes the conformation of the active site, no more of that production, this production loop is stopped, right? There's nothing more happening. That production loop is stopped. As the concentration of AB is reduced in the blood, this one leaves the system. Whatever was attached to this allosteric site leaves the system and then goes and produce and is involved in whatever activity it needs to be involved in, and then the active site, the shape changes. Once the active site's shape changes, the production line begins again. So the end product of the feedback loop itself acts as an inhibitory system. Just to recap this, if you have 5 steps to produce the product AB, when the concentration of AB in the blood increases, in the system increases, the product AB itself will go and attach to the allosteric site of the enzyme and then change the shape of the active site. And once it changes the shape of the active site, there's no more substrate, the substrate cannot attach to it, and the loop is, and then that chain is broken. And that's a negative feedback loop. In the case of prokaryotes, because they do not have membrane-bound organelles, you're going to find various enzymes in the cytoplasma, whereas in the case of eukaryotic cells, there are very specific enzymes that are found in very specific locations. They tend to for example, in the mitochondria, you're going to find a specific types of enzymes in the mitochondria. In plant cells, in the chloroplast, you're going to find specific types of enzymes involved in production there. So there is a certain level of specificity for enzymes. If you think about your digestive system, salivary amylase that breaks down carbohydrates is found in your saliva and in your mouth. Pancreatic amylase that breaks down carbohydrates further is found in your duodenum. Your pancreas pumps it and goes into your duodenum. Pepsin is found in your stomach because pepsinogen requires a low pH to be activated and become pepsinogen. So that is only found in your stomach. That is a very low pH environment. Trypsin is found in your pancreas, trypsinogen is pumped from your pancreas, and then once it enters the duodenum, which is the top part of your small intestine, then it is activated. So different parts of your body have different types of enzymes that do specific jobs that need to be done in that area. So with this, we kind of end this chapter. I'm going to start chapter 9, which is cellular respiration. We're just going to do basics of cellular respiration right now, and then we'll finish it up in class. Cellular respiration as a chapter is going to be very heavy on chemistry, so kind of brush up on your macromolecules. Just chapter 9. Everything that is living needs to acquire energy from some sort of an outside source, whether it is ingestion, whether it is absorption of macromolecules directly. However they do it, every single organism needs to gain energy from an outside source. So within our ecosystem, energy is basically flowing from the sunlight. From sunlight, it gets to plants, and then plants to it gets to animals and other organisms. So photosynthesis, the equation for photosynthesis is CO2. Carbon dioxide is taken in with water in the presence of sunlight. It makes C6H12O6. So glucose is produced by plants. So this process would be an anabolic process because it is making something. And O2. And this is done in the organelle chloroplast. And then animals and plants themselves also use it, are consuming this exact same, this product. So this reaction goes in this direction and then produces CO2 and H2O. And that's that. And along with that, it produces ATP. So when we say they produce ATP, what we actually mean is that it regenerates ATP. It produces the energy to do this reaction. It produces the proton motive force, the electrochemical gradient required to do ADP plus PI to make ATP. Depending on the type of reaction, the type of respiration we're doing, it's going to go anywhere from 4. If it's doing just fermentation, then it is 4 ATP per glucose molecule. If it does aerobic respiration, then it is doing up to from between 36 and 38 ATP per glucose molecule. So it is exactly the same reaction. A reaction going in this direction is photosynthesis. The reaction going in this direction is cellular respiration. Here's a good image of what it looks like, the 2 organelles involved in this process. Remember, both these organelles are considered to be a part of the endosymbiotic theory, which is they used to be prokaryotes that were ingested but not digested. So they have their own DNA. They have their own DNA, which is circular like a prokaryotic DNA. They do produce some of their own proteins. They do have some of their own ribosomes and everything. This reaction of making ATP is also, it's basically regenerating ATP. So the reaction actually is ADP plus PI. So once the ATP is regenerated, then your body uses it. It's stored energy, then your body uses it, and then it breaks down to make ADP plus PI, and we talked about it where it can go further down to make AMP, and then that AMP can become cyclic AMP and act as a secondary messenger for amplification of signals within your system. process of breaking down organic molecules. So a catabolic process. Photosynthesis would be an anabolic process, whereas cellular respiration is a catabolic process. This catabolism means to break down. This catabolic process of breaking down is an exergonic, also an exothermic reaction. That means it gives out energy at the end of it. That energy is used to make ATP. So there are multiple ways in which the cellular respiration takes place. At the end of the cellular, so there are multiple steps to it. At the end of the cellular respiration process, electrons are generated. So oxygen acts as a terminal electron acceptor. What do we mean by that? Oxygen is a very, very electronegative molecule. So that it is, what does electronegativity mean? Electronegativity is the ability of an atom to pull an electron towards itself. So since it is very electronegative, it is able to pick up the electron that is generated in this process. As we go along, one of the ways for you to do this is to actually follow the electrons. And if you follow the electrons, this chapter will be very, very easy. Just try to figure out where the electrons are being generated, where they're giving out their energy, and what is picking it up. If you just follow the electron, it'll be like a really easy time. So there are multiple types of respiration that takes place. The first one is aerobic respiration. Aerobic respiration is when oxygen is involved in it. The terminal electron acceptor is oxygen. Under aerobic respiration, there are two different types. One is obligate. And then the other one is facultative. And I will get to that in what it means in a second, facultative. The second type of respiration is anaerobic. Anaerobic. Under anaerobic also you have the same things. You have an obligate and a facultative. So what does it mean to be an obligate aerobe? An obligate aerobe is an organism that can only use oxygen as its terminal electron acceptor. An anaerobic organism is an organism that uses anything but oxygen as its terminal electron acceptor. It can use hydrogen sulfide. It can use the different components of nitrate. There can be oxygen with something else, but not oxygen by itself. It can use nitrates. It can use carbon dioxide. A whole bunch of different chemicals can be used as terminal electron acceptors. Can use anything but oxygen. So if an organism is an obligate anaerobe, that means that organism can only use some other chemical besides oxygen. It cannot use oxygen. Now in the middle of these two things are things called facultative, the aerobes and anaerobes. When oxygen is present in abundance, then it will use oxygen as a terminal electron acceptor. If oxygen is not present in abundance, then it will use whatever is available or whatever it can use. So within aerobic is when oxygen is used as a terminal electron acceptor. Anaerobe is when oxygen, anything but oxygen is used. Obligate, aerobe or anaerobe, just follows whatever. There's no leeway there. They just have to use what they have to use. Facultative is depending on the situation. Aerobic respiration is much more effective. It produces anywhere from 36 to 38 ATP at the end of the process of catabolizing one glucose molecule. Anaerobic produces anywhere from 26 to 28. So if an organism has an opportunity to choose between being aerobic and anaerobic, if it's facultative and oxygen is available, then it is just going to do aerobic respiration. It is not going to do anaerobic respiration. Another thing to remember in this is eukaryotes are only capable of doing aerobic respiration. They are only capable of aerobic respiration. Most anaerobic respiratory organisms, we cannot say all because, you know, you haven't like found every organism that is there on Earth. Most of the anaerobes, facultative anaerobes, are all in the family of prokaryotes. A good example for a facultative aerobe and anaerobe is E. coli. E. coli, when it is in your gut, does anaerobic respiration. In other places, if oxygen is available to it, it will do aerobic respiration. Both of these, between aerobic and anaerobic, there's something called as fermentation. Fermentation is basically a partial degradation of glucose. When oxygen is not available, fermentation takes place in an aerobic organism, or whenever the terminal electron acceptor is not present, a part of the degradation of sugars are called fermentation. The objective of fermentation is to regenerate NADH, which is a molecule that carries that is capable of carrying a high energy electron. It's capable of attracting that high energy electron and carrying it across the membranes to different areas to do different jobs. So fermentation's job, when oxygen is not present, is to do a partial degradation, produce some ATP without producing, and not producing all the ATP that it can potentially produce, but also regenerating the molecule NADH. There are two different types of fermentation, and then you guys are probably aware of. One is alcohol fermentation that produces beer and all the other alcohols. The other one is lactic acid fermentation that if you work out a lot and, you know, if you're running really fast, you get that sharp pain in your gross muscles. That is lactic acid fermentation. We will look into it more in detail in the next coming slides. Just talking about it, which is cellular respiration, basically includes both of these things. You are breaking down carbohydrates so you're breaking down this with the help of this as the terminal electron acceptor. This is what is regenerated. This is what you breathe out. Energy is produced, and that energy is used to make ATP, which is regeneration of ATP. Please remember that part. I'm gonna end this here, and then we'll do this part of NADH and what it does and how it works in class, just in case you have a lot of questions. This can get a little confusing. So see you guys.